Hi friends, my name is Nandita. I am from Andhra Pradesh.
Now let us discuss about the seed. Seed is a ripened ovule which contains an embryo or tiny plant with sufficient reserve food for the development of embryo. The ovules after fertilization develops into seeds. A seed is made up of seed coats and an embryo. The embryo is made up of radicle, an embryonal axis and one or two dicotyledonous. Types of seeds. Seeds can be divided into two types based on the number of cotyledons, presence or absence of endosperm, that is dicotyledonous seed and monocotyledonous seeds. Now let us discuss about dicotyledonous seeds. Gram seed is a dicot seed formed in a small pod or legume. The outermost covering is the seed coat and endosperm is absent. Seed can be situated under two heads, that is external structure and internal structure. It is light or dark brown in color. Its surface may be smooth or wrinkled. A small ovule scar present at the side called helium. It is the point where the star called funicule of the seed is attached to it. A narrow ridge called raphe runs from helium to chalaza inside the furrow. A small pore called micropyle present between the helium and pointed end. The outermost covering of the seed is seed coat. The outer hard and the leathery layer of the seed coat is called testa, and the inner thin and membranous layer is called tegma. In some seeds, the tegma and testa are fused. The seed coat encloses the which is differentiated into the radicle, a plumule and cotyledonous. The radicle develops into root and the plumule into shoot, and cotyledonous may be one or two to serve as reserve food. Hypocotyly is a part present between point of attachment of cotyledon and radicle. Epicotyl is present between the point of attachment of cotyledons plumulae. In some seeds, such as castor seeds, the endosperm is formed as a result of double fertilization, which is a food storing tissue. In some plants like bean, gram and pea, the endosperm is not present in mature seeds, that is non-endosperm seeds. Now let us discuss about monocotyledonous seeds. The monocotyledonous seeds are endospermic, but some are in orchids are non-endospermic. In the cereals such as maize, the seed coat is membranous and generally fused with the fruit wall. Let us discuss the structure of monocotyledonous seeds. In monocotyledonous, the endosperm is bulky and stores food. The outer covering of endosperm separates the embryo by proteinous layer called alurone layer. The embryo is small and situated in a groove at one end of the endosperm. It consists of one large shield-shaped cotyledon known as scutellum and a short axis with a plumule and a radicle. The plumule and radicle are enclosed in sheets which are called coleoptile and coleorhiza respectively. The coleoptile has a terminal pore for the emergence of first leaf during germination. The sheet is capable for growth. It helps the future shoot in passing through the soil during germination. Example, maize grain is whitish yellow, violet or red in color. It has smooth or shiny surface. Its grains is covered with a single thin hard covering. It is formed by the fusion of seed coat or testa, and the fruit wall is called pericarp. Now let us discuss about various morphological features are used to describe a flowering plant. The description has to be described in a simple and scientific language and presented in a proper sequence. The plant is described beginning with its habitat, vegetative characters, root, shoot and leaves, and then floral characters, inflorescence and flower parts. After describing various parts of a plant, a floral diagram and flora formula are presented. Thank you.